Dear students of 12th Medical, today in Botany we will continue our discussion on the Mendel's work, his experiments, and why his work remained unnoticed, and then the rediscovery of Mendel's work. As you must be knowing that the Mendel's work remained unnoticed for quite some time. It was a period of almost 34 years before his work was recognized. It was because of the limited circulation of proceedings of the Brunn Natural Science Society in which his work was published. And second, he could not convince himself about his conclusions being universal, since Mendel failed to reproduce the results on hawkweed undertaken on the suggestion of Nigeli. When Mendel was asked to repeat his experiments on the hawkweed, his results were not the same as were published by him. And third, lack of aggressiveness in his personality. Number 4, the scientific world was being rocked at that time by Darwin's theory of evolution, the origin of species in 1859. And fifth, Mendel's concept of stable, unblending discrete units or factors of various traits did not find acceptance from the contemporaries, and Mendel's conclusions about heredity were ahead of his time, and he used statistical methods and mathematical logic which were unfamiliar to other biologists at that time, and there was no physical proof for the existence of factors or the material they were made of. This means that the Mendel's experiments were far ahead for the other living scientists then to accept his work, because nothing could be known at that time about the discrete particles which Mendel talked about, because nothing like that was previously known to the scientific world. So his work remained unnoticed for quite a substantial period of time. It is almost 34 years. And then his work was rediscovered. The rediscovery of the Mendel's work. And Mendel died in 1884, long before his work came to be recognized. It was in 1900 that 3 workers independently rediscovered the principles of heredity already worked out by Mendel. The principles were already worked out by the Mendel in his experiments on the pea plant. When 3 of the scientists rediscovered his work, they were Hugo de Vries of Holland, Karl Correns of Germany, and Erich von Schirmack of Austria. Correns raised the status of 2 of Mendel's generalizations to the level of laws of heredity, law of segregation and law of independent assortment, which we are going to deal in detail in the classes coming ahead. And the others are variable principles. Hugo de Vries also found out the paper of Mendel and got it published in Flora in 1901. And Bateson, Punnett, and other subsequent workers found Mendel's work to be of universal application, including animals. And now, the reasons for Mendel's success. What were the reasons why Mendel was so successful with his experimentation? Is Mendel selected only pure breeding varieties of pea, that is Passum sativum, for his experiments. He took 2 years, 1857 to 1859, for checking that his experimental material are pure breeding. And Mendel took only those traits for his studies which did not show linkage, interactions, or incomplete dominance. Characters chosen by the Mendel had distinctive contrasting traits like tall and dwarf, or green and yellow. And Mendel took on one or two characters at one time for his breeding experiments, while his predecessors often studied all the traits simultaneously. And Mendel studied the inheritance of a character for 3 or more generations. And he performed reciprocal crosses and raised large progenies. Mendel's experimental plant, that is P. passum sativum, is ideal for controlled breeding. It is crossbred manually, while normally it undergoes self-breeding. And he took care to avoid contamination from foreign pollen grains brought by insects. And Mendel kept a complete record of every cross, subsequent self-breeding, and the number of seeds produced. Mendel experimented on a number of plants for the same trait and obtained hundreds of offsprings. A large sampling size gave credibility to his results. His sampling size was as large as 10,000 plants. And he formulated theoretical explanations for interpreting his results. His explanations were further tested by him as to their validity. And Mendel used statistical methods and law of probability for analyzing his results. And Mendel was lucky in selecting those traits, the genes of which did not interact, and they were either present on different chromosomes or showed complete recombination. He did not combine pod shape and plant height in any of his dihybrid crosses, the genes of which are close together on chromosome and do not show frequent recombination. And he did not attempt to explain all the variations found in his results, but left them as such. For example, linkage of flower and seed color. Now, in our next class, inshallah, we will take up what is prescribed in our syllabus, that is the Mendel's laws of inheritance, inshallah, in the coming classes. Thanks. Have a nice day.